read the gene it's supposed to.
So really there wouldn't be the rest of the strands.
Right, so you could just pull all of this away. When you're in here working, separate those two strands out to be able to get into those bases. Otherwise, those bases are going to be hydrogen bonded together and you can't get to them. So we're not going to use a helicase to unwind the entire thing, but we will actually pull the strands far enough apart to be able to get to one side of it. The other side will not be involved at all. Only read one side to do this.
What's the function of the transcription factor again?
So the function of the transcription, there's a lot of functions to the transcription factors that we're glossing over. For our class, what we want you to know is that's what's binding to the promoter site and providing a site for the RNA polymerase to bind to.
Sorry, I almost forgot what I was going to ask. So, does the RNA polymerase like overlap that whole area, or does it just, like, how much would it actually cover?
How far is the RNA polymerase actually overlapping things?
Yeah, like, does it go all the way to the gene, or?
I'm not sure how far it goes. I know it's bigger. I don't know exactly how far it overlaps. Yeah, I don't have a good answer for that. I'm sorry.
Okay.
Yeah.
This is literally just repeating what you said, but the promoter regions are the binding sites for the transcription factors, yes?
Promoter regions are the binding sites for the transcription factors.
Okay, cool.
Transcription factors are then the binding site for the RNA polymerase. Yeah. Yes.
Does RNA polymerase read?
Thank you. That's where we need to go next. So when we talked about DNA polymerase, the other thing that we mentioned was that it could only read in one direction. We said that DNA polymerase read 3' to 5'. Turns out that's just how DNA gets read. So you're reading DNA again. This RNA polymerase can also only read 3' to 5'. When it synthesizes RNA, it will make that complementary strand 5' to 3'. So since we were reading this way, genes down here, we know this has to be the 3' end here. This is the 5'. So that was 3, and what I erased up here was 5. The new RNA strand that we're making is going to be 5', 3'. And again, it's going to be complementary. So this would be G, A, C, and G. Basically the same thing as making a complementary strand with DNA, but just take all the T's and replace them with U's. Okay. Yes?
Does RNA distinguish, or I guess does transcription distinguish between the template strand of DNA versus like the new strand that was made? Or does it not have like a preference?
So when you're reading this and transcribing, does it matter which strand? Do they care if it was a template strand versus a new? No, it doesn't. You know, once they've become a DNA molecule, it's not really obvious which one's new and old. You'll methylate it, they'll look the same. The only thing that is going to matter is that it has to come in from a 3' to 5'. So if it had a gene here that it was trying to read, the promoter would have been back here, and that's where everything would start this way. Yes?
What did you say had to open the strand DNA? Was the RNA polymerase signaling helicase, or did the helicase just come by its own?
Okay, so the question is, how are we getting this DNA open so that we can actually read this? We don't use a helicase in transcription. We just use the RNA polymerase. Okay, it's a lot more complex and we're trying to gloss it, but there are proteins that will actually pull and hold this apart so that you can get all this stuff in here and read it. It will not be a helicase though. So if you see helicase, that's very specific to replication. In fact, that's a really good study tip is to go through and list transcription, translation, replication, and write all the words you know for each one of those, the vocab that goes with them. That way you're comfortable enough that as soon as you read helicase or you read promoter, you'll instantly know what story you're at. And if you're in a fill-in-the-blank question, you'll be able to think of all the words that will probably have to go in those blanks. Another question.
What exactly is the gene for? Like, is that just what they want to find here?
So what is the gene? So a gene is a portion of your DNA that actually holds a code that you want to express. This is the code for making a particular protein. So we'll have genes throughout our DNA that we can express into things that will function. They may just express into rRNA, not actually a protein. And then in between that, we often have junk or things like promoters and things that can help regulate how often we'll transcribe those genes. Yeah. Yes?
Is the RNA being copied from the strand with the promoter region or the transcription factor?
Okay, so the question is, where exactly are we transcribing? We are going to be transcribing the strand that has the promoter and the gene on it. So I tried to draw a double strand and then just erase the one side to show you, but I think that was pretty confusing. This is just gone now. We pulled it completely apart, and everything that's going on is going on this bottom strand that's going 3 to 5 prime. So you'll have the promoter, you'll have the transcription factors on top of the promoter, and then the RNA polymerase on top of those transcription factors, and the whole thing, that whole polymerase now can move down and read the gene and transcribe it. Yeah.
What is the factor that pulled them apart, the strands that it could read?
We don't say.
Oh, like.
We don't tell you that, no.
Okay.
So what's pulling it apart? We typically tell you that the polymerase is doing that. It's more complicated, but we're not going to ask you what pulls that apart. Yeah. Okay. All right. So transcription, it's not that different from replication, just where you're going to start and then the different bases that you're going to use. Of course, it's going to be much shorter than if you were replicating an entire genome. All right, so now that you have made this RNA, this is not a final RNA. What you've made here is actually going to be RNA. Which brings me to another point. So we know that DNA is always going to be in the nucleus. It stays in the nucleus. So anytime we're reading or working with DNA, we have to be in the nucleus. That means replication occurred here. It also means transcription occurs there. But in order to actually make our protein, synthesize our protein, we have to move it out into the cytoplasm. Before we could do that, we're going to take this pre-RNA and edit this in the nucleus. Okay. I'm going to draw this like a box. This could be an mRNA, but this could also be an rRNA or tRNA. It's any RNA. This is going to have a series of sequence bases on it. That are, again, consensus sequences. So remember that a consensus sequence is just something that's conserved, meaning that that series of bases will do the same thing. That doesn't mean that all consensus sequences are the same. So this is necessarily TATA, it probably isn't. TATA is a promoter. This is going to be a cutting and splicing site. So we have these other proteins. You don't need to know this, I'm just writing this down because it sounds weird when I say it. It's a SNRP. It's a small nucleoriboprotein, something like that. It cuts your pre-mRNA. So it looks for, it will have the matching complementary bases to these consensus sequences, and that tells it to come in here and cut. So what we could have had, this could have been exon 1, this could have been an intron, exon 2, an intron, and exon 3. So when the SNRP comes in here and it cuts this, you can actually remove your introns. Okay, this one and this one will be discarded. And now you could put those 3 exons together. So the SNRP will also, what's called splice. So cuts it out and then puts it back together. So this would have been, I'll just say E1, E2, and E3. So now we've gotten rid of those non-coding regions and we've just left behind the stuff that we actually need to work, to make our protein, to make our functional rRNA, tRNA. But what's really cool about this process is that it doesn't always have to be this series. You could also cut those out, but then only take 2 of them. Say we'll do E1 and E3. This could be E2 and E3. Maybe you drop them all except E1. You can combine these, mix and match these exons in different ways. Not all of them will be functional. Not all of them will happen. Okay, in this particular gene, E1 and E3 may not do anything. May not even be spliced ever. But by being able to have at least a couple of mix and match options for some of these genes, it means that we can have just 20,000 genes. I can't believe I said just 20,000. But we can have 20,000 genes but make so many more proteins from it than that. We're not limited because we can now make protein 1, protein 2, protein 3 from the same gene. Yes?
So I just wanted to clarify, for the pre-RNA, you said it could be any type of RNA? So it'd be like pre-tRNA, pre-mRNA?
So the question is this pre-RNA, I said that it could be any type, and that's true. So this could be what will be an rRNA, an mRNA, a tRNA. We tend to explain things in terms of mRNA. So like this is more mRNA because it's the mRNA that's our blueprint for proteins. Able to get the different proteins from it. If you're making a tRNA, you have to have the certain exons, whatever those particular exons are for that gene to function. So you don't get the mix and match with a tRNA, but you will have to edit all of your different RNA before you can send it out.
So does it have to be exon, intron, or can it be like really any order?
So the question is, can it be any order? That's a good question. I was going to say, well, how would you know they're different? But you could still have splice sites. I don't know the answer. Yes?
Could one section of pre-RNA code for, or turn into, I guess, different types of RNA after splicing out? Like, could you use exon 1 and 2 and have it be mRNA and use exon 2 and 3 and have it be tRNA?
Okay, so the question is, can you edit this in such a way that you would actually get different types of RNA? Could, if you had E1, 2, and 3, this have been a tRNA, but if you make it E1 and 3, that actually could be an mRNA? I don't think so, primarily because we actually use different polymerases to make these different RNAs. So that was kind of another detail we gloss over for you, but I suspect that would prevent being able to make multiple same editing process. Yes.
So is this now with the exon spliced together, is that like the regular RNA now?
So this is now final RNA, whatever type it is. Yes. Yes.
So the exons have to 1, 2, 3, or 1, 3, even like after splicing?
So do the exons have to be in order, or can we actually have, you know, a factorial, I don't, math is not my thing, sorry, but can we also start like flipping these and have E1, 3, E2 for another protein? Probably. Yeah.
Another question. Can like one prion splice into multiple different final RNAs, or does one pre-mRNA just become one final?
So would one pre-mRNA become one final, or could you make multiples? Like, could you do an E1 and 3 for one mRNA and then have your leftover E2 as the second mRNA? Yeah, you can do that. Okay, you're not going to duplicate them, but you can use all your exons. I think that one was.
What happens to the introns once they're
So what happens to the introns when they're discarded? These are nucleic acids. Okay, and we don't keep nucleic acids around very long. We have nucleases that will break them down back into their individual components so that we can put them back together again as new. We just recycle them essentially. Yes?
What is the difference between like an exon and an intron, besides how they're kind of spliced out or just are together?
So what's the difference between the exon and intron? I mean, they're all nucleic acids. They're going to look the same. They have the same base type. It's just the sequence. We'll have these consensus sequences that will signal that this is what you want to keep and this is what you don't. All right, so it's not that there's a difference. It's just that it's already tagged as to what we're keeping and what we're not, of what's tagged for discard.
Got it. Okay. So then are all consensus sequences tagged to be discarded, or are some kept and some are not?
So a consensus sequence can be a number of different things. It just tells you that that's a sequence that you will see used the same way over and over again. For the specific consensus sequences for splice sites, for cutting and splice sites.
Oh, I didn't. So the consensus sequence for like the promoter region is different than the consensus sequences for exons and introns?
These consensus sequences will be different from other consensus sequences like promoters and yes. Yes. Way in the back.
So for introns, why do we have them for sequences to our energy to transcript them versus discard them?
So I'm not sure I heard you. Ask again if I'm not answering correctly, but I think you were asking why do we have an intron if we're just going to get rid of it? Yeah. Okay, so these introns often contain codes that will have effects on how often this gene gets expressed. It may also be affecting to some the order that these are being put into. Okay, so they sometimes do have functions. Other times they may truly be junk DNA that we just don't use anymore. We've evolved beyond this particular portion, and it's really hard to just kind of get rid of that except over long terms. Survived just because we didn't need them, so. Yes.
Is it possible to have final RNA with introns still in it, or are the introns always taken out?
So is it possible to have a final mRNA with introns still in it? Possibly, if you have, say, a mutation in your SNRPs that aren't functioning properly, but then that would create a non-functional final RNA, or it would cause probably problems with it. It wouldn't function as well. Yeah.
I had written down while you were speaking, and you had said that the SNRPs remove the introns and put exons together, but then you wrote down the consensus sequence as splicing. So who was doing the splicing?
So the consensus sequence is the bases on the, that is the site that the SNRPs will come in and cut. So that's the site of cutting, but it's the SNRP itself that will actually do that cutting and splicing.
Okay, so it's a signal for the
Yes, it's the signal. Okay. All right, so now we have these final RNAs. If this was ribosomal RNA, we're also going to complex it with proteins to make the ribosome. But now all of these can leave through the nuclear pore out into the cytoplasm so that they can function. So if it's a ribosome, it's going to be picking up mRNA to read it. If it's an mRNA, it's going to get picked up by the ribosome. Let's actually review those RNAs, because we're going to talk about them in detail tomorrow. Okay, so rRNA, also known as ribosomal RNA. This will complex with proteins. And it's going to create a ribozyme that can read mRNA and catalyze peptide bonds between amino acids. We have our mRNA, that's messenger RNA. Okay, this is going to be the blueprint for our protein. We could talk tomorrow about how exactly we're going to use those 4 bases to code for specific proteins or specific amino acids to make that protein. But that gets read by the ribosome. Now, in order to turn this, translate this from the nucleic acid to that amino acid, you have to bring the amino acids in. And so we've got our tRNA. That's our transfer RNA. This is going to shuttle amino acids to the ribosome. Okay, so we will walk through that step by step tomorrow to talk about protein synthesis. We'll talk about both free and we'll talk about the bound protein synthesis. The last RNA I want to leave you with is not one we're going to talk about. I don't know if you can read it down here, but it's regulatory. So it's actually very important. It just doesn't fit into the story well, so we kind of gloss that. As you'll get this when you talk about genetics, maybe some biochem. Regulatory RNA is really important in regulating your other RNAs, when things turn on and off, when things are degraded. But it doesn't necessarily play a big part in the protein synthesis as we tell it, so that's all we'll talk about it. Yes? So can I explain tRNA again? This is transfer RNA. It's an RNA molecule that will actually carry an amino acid to the ribosome to be added to the protein. It will have to match acid to the correct sequence basis on the RNA. And we'll talk about how it does that tomorrow. Yeah?
So is that RNA just bouncing around, or is that using a certain cytosol?
So is tRNA just diffusing, or is it using cytoskeleton? It could potentially use both. It could do both, yes. Yeah. All right, thanks, guys.